Hello, and welcome to today's virtual overview of what often happens when fish are submitted to a diagnostic laboratory for fish health assessments. Because we have a diverse audience tuning in, our hope is to give folks from a range of backgrounds just a taste of the array of things that go into the clinical examination and laboratory analyses of fish, using my lab, the MSU Aquatic Animal Health Laboratory, as an example. For those of you that don't know me, this is my mug, and my name is Dr. Thomas Locke. I'll be narrating this tour today. So before virtually introducing you to the amazing AAHL crew, I want to preface this tour by saying that in my lab, the majority of the analyses we do are in keeping with the recommendations put forth by the American Fisheries Society Fish Health Section Blue Book and/or the World Organization for Animal Health, or OIE. So if you're interested in further detail on some of the procedures that you'll get brief glimpses of today, those are excellent resources to consult, and I'm always happy to answer questions using my contact info that you can see here. So next, it's my pleasure to virtually introduce you to some of the MSU Aquatic Animal Health Lab crew that are the heart and soul, or really the gills, heart, and liver of the lab. They're truly instrumental in everything we do here, and also were a huge help in preparing this virtual tour. So I've listed their names in order of appearance here, just like they do in the movies. So, of course, while I have a second, I also want to give a huge thanks to Elliot Nelson for his video and recording skills in preparation of this tour. So as I mentioned, my labs are on the campus of Michigan State University, where I'm the principal investigator of the MSU Aquatic Animal Health Lab and director of the Aquatic Animal Disease Ecology program. Currently, my lab accepts submissions and samples from state, federal, and private hatcheries and aquaculture facilities. Fish are most often submitted for either health certification or for determination of what's behind a mortality or disease event. Either way, the submission process begins by contacting my lab manager to schedule a submission date and time, and then filling out the appropriate paperwork that we use to collect some initial case history information. The initial connection between the submitter and the lab is really critical towards ensuring that adequate preparations on the lab side can occur. Once submission arrangements have been made, fish are usually submitted freshly dead on ice, and if a submitter is ever unclear on best practices for this, one should work with their fish health lab to ensure samples arrive in a condition that's adequate for the required analyses. But in brief, plenty of ice and layering it around thin layers of fish rather than a mass of fish being in one bag is usually preferred. In certain cases, some labs, including mine, prefer that live fish be examined, as this affords the opportunity to observe fish behavior and provides optimal conditions for several additional analyses, like blood collection and histological analyses. So my lab's equipped with aquaria where fish can be maintained for such purposes, as seen here. Once live fish have been observed and euthanized, or when receiving freshly dead fish on ice, the next step is to collect basic morphometric data, including measuring the length and weight of the fish. After morphometric data has been measured, and so long as the fish are very freshly dead, blood can then be collected and used for a variety of purposes, including assessments for current and previous infections, red and white blood cell counts, and others. So to do so, sterile syringes and needles are often inserted near the base of the tail, where blood can then be collected from the vessels that lie ventral or just underneath the spinal cord of a fish. Once collected, the blood can be used to make blood films for microscopic examination, for viral or bacterial culture, and it can be spun down in a centrifuge and the resultant serum used to look for antibodies that indicate previous infection. Depending again on the purpose, you can make skin and gill preparations or whole mounts, or like basically take tissue biopsies and look at them under the microscope for external parasites or certain bacterial infections. So normally what we do for this, for a skin biopsy, a lot of the parasites that we want to look for, a lot of times they like to hang out behind the fins, and so the mucus is like you're basically trying to get a mucus sample. So we like to use these clear plastic cover slips that can bend. And so basically what you're doing is you're just trying to like gently
get a representative sample from like across the surface of the fish. So you can see, you know, like we got a little bit of mucus there, right? Okay, and so from there what we do, get that down on the slide and spread it out. If you do it right, it should, yeah, like I can see that that's like a bit opaque. So basically we've now spread out that mucus, and there's a few scales in there, and then we'll be able to throw it on the microscope and look for parasites. So that would be what we would consider like a skin preparation.
Okay.
All right, so then what you can do is, if you want to look at the gills, so there's a lot of different ways you can do this, but I'm just going to cut off the gill cover or the operculum of the fish here to give us better access to the gills. Oops. I'm getting a little aggressive, but it's all right. Okay. So now we've exposed the gills here. So I'm going to basically take one of these hemibranchs and just like excise it. So just a couple snips, and I'm going to put that right on the slide. Now this is too much. Like if we were to put a cover slip on there, that cover slip is not going to go down.
Okay.
So what I'm going to do from here is use like a nice fresh scalpel. It's very sharp. So I'm just going to very gently excise some of these gill lamellae, like the finger-like projections, right into that solution that I put on the slide. Okay. And that way, then I'm going to throw another cover slip on. And this lets us look not only at like the structure of the gills, but also if there were any parasites that are present, or bacteria for that matter.
When looking at the skin and gill preparations under a microscope, we may encounter a wonderland of parasites or bacteria. So here we're providing an example of healthy gills, along with several examples of protozoan and monogenian parasites that one might encounter. Of course, the type and number of parasites will really determine what treatment action, if any, would be undertaken.
For every single fish, like normally we would get, say, 60 fish or 120 fish or something. Each fish is going to have its own separate tools, and because obviously we can't be cross-contaminating between fish, right? Mm-hmm. So everything is very compartmentalized. Like this is our fish one, so we have one set of tools for this fish. So after we've collected blood and done our whole mounts, then what we do is we like give the fish a quick exam. So we're looking for any external signs of disease. So like we're spreading the fins out. We're not seeing any signs of disease, so we're giving like a quick look at the fin margins. We can look inside the mouth to see if we're seeing anything of concern. So we can give it, sometimes there's parasites that really enjoy hiding in the mouth of the fish. We can look at the gills, and this can tell us a lot. We also look at the eyes of the fish to see if there's anything of note. We look at the nares of the fish. Sometimes we can start to see erosion around the nares or the lateral line of the fish here. Also take a quick peek at the vent of the fish. So this is like the cloaca. Sometimes that can be inflamed if the fish is suffering from a systemic disease. And then of course we don't just want to look at one side of the fish. So we're going to look at both sides to see if we see anything of note. You can see here a little bit of congestion at the base of the fins. So once we're done with the external exam, then what we like to do here is we like to spray the fish with 70% ethanol because there's bacteria that live on the surfaces of the fish. And now that we're going inside the fish, we're really interested in if there's an infection inside, we want to know. And so we don't want to contaminate the insides of the fish with the outside. So that ethanol helps us reduce the chance of that. So from here, I'm going to use scissors, and I'm going to just start to make a small incision just ahead of the vent here. I'm going to make one more small opening, and basically I just want to give myself access to all the visceral organs. So I'm going to cut up through this branchiostegal arch here. I'm going to come back. Oh, you can see this fish just ate semi-recently. Well, very recently. So I'm just going to finish the cut. So basically here, I'm looking for general signs of disease. I can also get a sex of the fish. So like here, these are the gonads. So even though this fish is pretty immature, I can see, looks like this is a male. These would be testes here. So I would note that. So this is a male. And from there, I would start to note things like how much fat does the fish have. So like we give fish a visceral fat index, or VFI. That's a scale from 0 to 4.
Mm-hmm.
I'm going to say this is like a VFI of 2. From here, I'm going to start to collect the tissues for virus isolation. So this is the spleen of the fish. And so I'm going to take a small piece of the spleen. And I'm going to place that carefully in the Whirl-Pak. If you notice, like I have two forceps. This is the one that I'm really using to touch the outside of the fish, and this one I'm really only using to collect my samples. So I'm saving this for like the sterile type of stuff. So we got our spleen. This is the heart of the fish. So they have sort of a four compartments to their heart. They've got a sinus venosus, then this is the atrium, ventricle, and then that goes to the bulbus arteriosus. Yeah, so I took a small piece of the heart. You know, I like to pull the heart out, and we can also get a better look at that to see if there's anything of concern. So next up, we look at the liver of the fish. So this is the liver. That actually looks pretty good. Below the liver, you can get a look at the gallbladder. So this is the gallbladder here, this yellow like bulbous structure. And that looks pretty normal. So it's yellow and a bit distended, but it's because the fish is actively eating. So here, this is the stomach, and you can see these little like knobby-like structures in the stomach. This fish is being fed a pelleted diet, and so these are actually undigested pellets that we're seeing in the stomach. So I like to sort of flip the gastrointestinal tract over so I can look at all the sides of the fish, the GIT. So the stomach winds around to the intestine here. So this is the intestine, and that has a lot of feces in it. So you can tell that this fish has been very actively feeding. And that color of feces is very consistent with what you would see in a fish being fed an artificial diet. So all in all, the fish looks pretty darn good thus far. Next up, we would want to look at the swim bladder of the fish. So I'm gonna just sort of get a quick peek in there, and that actually looks nice and healthy. So no concerns there. So then I'm gonna use the back of my forceps and just sort of gently scoot away the swim bladder here. And that exposes the kidney. So this entire structure here is the kidney, all along here. So fish are unique in that they don't have bone marrow. So a lot of the tissue that creates or generates red blood cells and white blood cells is all in the kidney of the fish. So this is very much like bone marrow consistency, but this is part of that's the kidney. So the kidney is also an excellent place to look for bacterial infections, because this is a place where a lot of bacteria will, if a fish recognizes something as foreign, then that's where it's going to be brought. So from here, what I'm going to do is I'm going to take a sterile loop, and I'm going to just run that back and forth along the kidney. So I'm taking a small inoculum. These are like calibrated loops. And I'm basically going to start streaking this tissue out onto different media that will allow different bacteria to grow. From there, then, I'm going to take some kidney tissue. So even though this looks like one, it is all kidney, but they sort of have a gradation of like a lot of immune function more at the anterior part, and then more excretory function at the posterior part. So I'm going to take tissues from both areas, because certain bacteria, and depending on the source, that can there might be bacteria in one region and not the other. So I'm going to basically take that, and then just like Megan was saying, so a lot of viruses and bacteria are very particular about temperature. So I'm going to scoop the tissue down, and then I'm going to close this up, and right away I want to get it on ice. So that way this sample is staying nice, and we're making the pathogens comfortable before we do all the rest at the food safety board.
Nice.
Okay, so that's that. In some cases, microparasites, like the causative agent of whirling disease in salmon and trout, which is Myxobolus cerebralis, may be suspected or need to be screened for. Although there are many methods to do so, here we're collecting cranial tissues to look for M. cerebralis in two different ways. So first, we're collecting half the head that will go on ice, and later, the skin, muscle, and other tissues will be artificially digested, leaving the cartilage and bone, which is then further digested to screen for parasitic spores under the microscope. The second half of the head is placed in a tissue fixative, in this case 10% formalin, which preserves the tissue and then allows for further analysis to not only further confirm a microparasite's identity, but also to assess any tissue damage that may be associated with its presence. So here's a little more on how we look for tissue damage at the microscopic level, or what we call histopathology. So here you can see a section through a fish head that's been preserved and embedded in paraffin. Then very thin, like 5 to 10 micron thick tissue sections are shaved onto a slide, stained, and then we can examine those under a microscope for tissue damage and for evidence of pathogens or parasites. So here we're showing an example of what the M. cerebralis spores look like in the cranial cartilage and bone of an infected salmonid. Now, as you'll recall, we collected kidney, spleen, and heart tissue as we were examining our lake trout earlier today, which we then placed on ice. Without freezing the sample, so we avoid negatively affecting pathogen recovery, we then process the tissues so that they can be used to detect certain bacteria or viruses of interest. Here, Sean is weighing the collected fish tissues, adding solutions at the target weight to volume ratio, homogenizing the tissue mix to enhance the recovery of any target pathogens, and then heating and centrifuging the sample so it's ready for enzyme-linked immunosorbent assay, or ELISA, testing, in this case for the bacterium that causes bacterial kidney disease. Aren't you totally impressed with how fast Sean is? I'm kidding, but next, Sean is processing a separate aliquot of the same tissues so it can be screened for the presence of infectious fish viruses. But more on viruses in just a second. Now, I mentioned ELISA testing for the bacterium that causes bacterial kidney disease. Because this bacterium grows very slowly in the lab, we use ELISA to detect this microbe more rapidly, and it lets us screen many samples simultaneously in a high throughput fashion. So here, Michelle is partway through these analyses, where sample wells are being rinsed, requisite solutions are added, and then the plate is incubated. After several more steps, the plate is placed in a spectrophotometer, which will detect color changes that result from the bacterium's presence, and then is interpreted via computer software. So again, this allows us to rapidly detect this pathogen, and in fact, we use this test to help guide the selection of eggs and milk that are free from infection to avoid disease in the resultant offspring. Okay, so coming back to how we look for viruses in the lab. This can be done in several ways, but here Dr. Chevalier is using the gold standard method of virus isolation via cell culture. Of note, and depending on multiple factors, it's not unusual for completion of these analyses to take 28 days or more. So here, and now that we've determined which viruses are suspected and which fish cell lines will be used,
Megan goes through the lengthy and technically rigorous process of culturing immortal fish cells in the lab. So remember that viruses require a living cell, in our case a fish cell, to replicate. So Megan capitalizes on this fact by growing fish cells in the lab, being sure to provide them with all the nutrients, growth factors, and substrates they need to be happy. She makes it look easy and fast in this video, but after numerous steps, Megan eventually propagates these fish cells in plates that are incubated at multiple temperatures. Next, a portion of the tissues that you saw Sean processing are inoculated into the wells that now contain these cultured fish cells, and the cell and process sample mix are placed back in incubators. Then these cells are incubated, often for up to 28 days, and periodically removed from the incubator to be examined under an inverted microscope. What Megan is looking for here is any evidence of damage to the cultured fish cells, or what we fancily refer to as cytopathic effects, because it turns out that when viruses infect cells, they also cause various types of damage that a trained eye can see under a microscope. So here, we're showing you an example of healthy cultured fish cells on the left, contrasted with cells showing severe lysis on the right, in this case as a result of a fish sample that contained viral hemorrhagic septicemia virus, or VHSV. Although some viruses cause fairly characteristic changes in cultured fish cells, when we see cellular changes like this, we then undertake additional lab tests to confirm the virus's identity. But more on that in a second. While viral analysis is occurring, so too are analyses to detect bacterial infections in our examined fish. Recall the plates that we streaked kidney tissue on? Well, those culture plates are incubated at multiple temperatures, again depending on the fish species and suspected bacteria that may be present. And then those plates are read periodically over multiple days. Here, soon-to-be Dr. Chris Knupp is reading a set of culture plates from a fish case and recording all bacterial growth that he sees. The idea here is that if we use the right growth media and incubation conditions, and if a bacterium was infecting the fish we analyzed, that same bacterium will grow and be present on these plates. When growth is present, we then subculture those bacteria onto new plates so that we can begin the process of bacterial identification. Once the bacterial subcultures have grown, an array of biochemical tests are performed, the results of which are then used to steer us towards bacterial identification. I told you Sean was fast. But in seriousness, we use results from these and other biochemical tests to navigate in multiple dichotomous keys, which then give us a presumptive identification for each recovered bacterium. If bacteria are recovered from an infected fish, we then undertake antibiotic susceptibility testing so that we can determine which, if any, antibiotics are capable of killing or inactivating them. This is critical data as it allows veterinarians to be more certain that an approved drug will in theory be effective at treating that particular disease outbreak, of course assuming the infections are occurring in a facility where such treatments can occur and in concert with veterinary oversight. Well, we showed you how we had suspect virus-positive samples and bacteria with presumptive identifications, but how do we get a final or confirmatory identification? Well, there are several methods for achieving this, but amongst the most common is by using molecular analyses like PCR testing. For this, we begin by extracting the DNA and/or RNA from our suspect or presumptive bacterial or viral samples. Here, Sean's showing you a very abbreviated overview of that process. Once the DNA or RNA has been extracted and quantified, it's then taken into a dedicated lab for molecular testing. In this case, we are using a PCR assay that is specific to the bacterial species we suspect our sample belongs to, based on the initial biochemical test results. Without getting into too much molecular detail, and if our initial results were correct, a part of one gene of this bacterium will be massively amplified as the tube and its contents are subjected to multiple reaction cycles.
Should that occur, and when we subject the sample to gel electrophoresis and view it under UV light, it will produce a band of a known molecular weight, or basically a known size. In this case, our initial results were indeed correct, and so we were able to confirm the identity of this bacterium as Flavobacterium psychrophyllum. Additional layers of confirmation can also be undertaken, such as through gene sequencing or genomic analyses. In other cases, we can use real-time PCR assays to confirm a virus or bacterium's identity, as Chris is showing you here. Such assays have the advantage of being fast, sensitive, and accurate, and can tell you how much of a particular microbe is present in a sample. Of course, along the way, and as all of the extensive clinical and lab analyses we covered today are completed, reports that summarize laboratory findings and subsequent recommendations, including any veterinarian recommended treatments, are sent to the submitter, with the goal of improving fish health in both the short and long term. Well, I know we packed a lot into this virtual lab overview, but my hope is that helps relay what often happens behind the so-called curtain in some diagnostic labs and conveys the degree of effort, time, and quality control that goes into a fish health submission.